Hello everyone, my name is Scott Hexmer from the Department of Physiology, and this video is going to focus on blood vessels and their role in cardiovascular physiology. I have nothing to disclose for these lectures. So our objectives for this video are to describe the determinants of blood pressure regulation and its physiologic relationship to flow and vascular resistance. By the end of this module, you should be able to describe the determinants of blood pressure and its relation to vascular resistance, and to understand the autonomic control of blood pressure regulation. When we speak about the vascular system within the cardiovascular system, we're really speaking about the plumbing, or the tubing through which blood is transported. Here we have a table of a number of the substances that are trafficked through the cardiovascular system. And what you really see from this table is how critical the optimized transport of blood and these materials is to the function of every cell in the body. When we start to understand more about this plumbing system, it's important to understand the anatomy of the pipes or the tubes that carry the blood and molecules. So in this image, we see the anatomy of a standard blood vessel. It has three layers: the intima, the media, and the adventitial layers. And these layers are separated by structural elastic laminae that help separate the individual layers. We look at the intima. This is really the lining of the blood vessel. And it's made up of one layer of cells called the endothelial cells. They're very tightly packed together, tight cell-cell junctions, and this layer really is a lining. It's to prevent materials in the blood from exiting the blood. If we look at the medial layer, that's really the wall of the vessel. And this is made up of vascular smooth muscle cells. This provides not only structural support, holding the vessel together, but it also provides a functional component, because these smooth muscle cells can circumferentially contract within the vessel wall to help the vessel change its diameter. And lastly, the adventitial layer is sort of the exterior coating of the blood vessel. It's made up of fibroblasts, extracellular matrix, and as you can see from the diagram, autonomic nerves interact with this area to help control blood vessel function. When we look at the overall schematic of the cardiovascular system, we really see a system of two pumps in series. And this is important to recognize as we try to understand how blood moves throughout the cardiovascular system. So if we follow the direction of blood flow from the left ventricle, what we see is that the blood moves into the aorta and then is split up to various pathways as it goes off and perfuses the different tissues of the cardiovascular system. As it perfuses these tissues, passes through their capillaries, you get exchange of CO2 for oxygen, and the deoxygenated blood is then trafficked back through veins and venules to the right side of the heart, or the right ventricle, where it is then pumped into the lungs for reoxygenation, and the circuit continues. In order to move blood through the cardiovascular system, you need to generate pressures, and these pressures are generated at the level of the heart. You've previously learned that the heart is a muscular pulsatile pump, and what that means is that it goes through periods of squeezing or systole and relaxing or diastole to generate these different pulsatile pressures. When we think about how pulsatile pressures relate to the pressures in the cardiovascular system, we can look to this graph at the bottom left of the slide to understand these gradients. So this graph shows us the pressure gradients as we move through the left ventricle, the arteries, arterioles, capillaries, and into the venules and veins. And what you can see is that there's a very large pressure gradient in the left ventricle between the systolic pressure and the diastolic pressure. So we've got systolic pressures of around 120 millimeters of mercury on average, and diastolic pressures around 10 millimeters of mercury. As we move into the aorta, this changes quite significantly. You can see that we still have systolic pressures around 120 millimeters, but the diastolic pressures are now at 80 millimeters of mercury. As you're watching this video, maybe take a pause and think about all the reasons why the pressures are so different between the left ventricle and the aorta. Okay, what are the underlying reasons for this? And once you get a handle on that, how might this ultimately relate to how blood is moved through the vascular system? Before we move from this slide, I want to address a couple other points. First, I want to talk about something called the pulse pressure. The pulse pressure is the difference between the systolic pressure and the diastolic pressure, and this is measured in the arteries. The other definition I want to give you is for the mean arterial pressure. The mean arterial pressure refers to the time-averaged blood pressure in the arteries. And you may notice that this time-averaged pressure, if you look at the solid line that follows the mean arterial pressure, you see that that solid line usually follows closer to the diastolic pressure than it does to the systolic pressure. It's not just the average between the two. In fact, it's closer to the diastolic pressure. The reason for this is because it's a time average pressure over the full cardiac cycle. The heart actually spends 2/3 of its time in diastole and only 1/3 of its time in systole, and so the mean arterial pressure then can be calculated by 2/3 the diastolic pressure plus 1/3 the systolic pressure. And if you rearrange this equation, as they often do in the clinic, you can also express it as the diastolic pressure plus 1/3 of the pulse pressure. So what is this interesting relationship between ventricular pressure and the systolic and diastolic pressure in the aorta, and how's it maintained? Well, we first get a clue when we look at what's happening right as the blood is pumped out of the heart into the aorta. So as the ventricle contracts, the semilunar valve opens and blood is ejected into the aorta. As it's ejected into the aorta, you may notice that the aorta stretches out. Okay, and that's actually what generates the pulse that we can feel on a patient's arm when we're taking their pulse rate. That squeezing, opening of the semilunar valve creates one common chamber between the ventricle and the aorta. And this is important because that means that any pressures that are generated in the aorta or in the ventricle are directly felt in the aorta as well. And that's why the systolic pressures of 120 are the same between the ventricle and the aorta. Think of it as one continuous tube. Now, if we look at what happens during ventricular relaxation, you see that the heart undergoes isovolumetric ventricular relaxation. That lowers the pressure in the heart and closes the semilunar valve. That closure of the semilunar valve, together with that elastic nature of the aorta, helps maintain the pressure within the aorta, and that squeezing elastic recoil provides an additional driving force for blood. The blood can no longer go back into the heart because the semilunar valve is closed and is forced or pushed along the vascular tree towards the various tissues. What we also know about the cardiovascular system as a whole is that the highest and the lowest pressures in the system are generated at the level of the heart. And so you can see in this diagram, if we follow the mean arterial pressure across the entire vascular tree, that our highest pressures on the left are generated just in the aorta, just outside the heart. The lowest pressures just in the vena cava, just ahead of the ventricle. And so one of the reasons for this drop in pressure across the cardiovascular system is that the blood experiences resistance to blood flow as it moves through the system. This resistance to blood flow is given by a law called Poiseuille's law. We'll study that later in the lecture, but at this time, you just need to recognize that as blood moves into smaller and smaller vessels as it gets further away from the heart, the resistance to blood flow increases. So there's a relationship between blood vessel size and resistance. The smaller the vessel, the higher the resistance, and this leads to this curve that we see for pressure gradients across the cardiovascular system. Now, it's key that that gradient be maintained at so that the blood pressure is higher at further upstream locations, as you'll see from the next slide. because pressure differences drive the blood flow through the cardiovascular system. So if we look at the hemodynamic properties of any blood vessel or tube, what we see is that the difference in pressure at two ends of a tube is what determines the amount of flow through that tube. So upstream pressures need to be high, downstream pressures need to be low in order for blood to move through a tube. If pressures are the same at both sides, there's no flow. And obviously that would be a disaster for the cardiovascular system. Now the relationship that determines pressure and flow is Ohm's law. You may remember Ohm's law from your work in electricity, and some of you may have had fluid mechanics before. But basically Ohm's law states that the difference in pressure at the ends of the tube is proportional to the amount of blood flow, and it's proportional to the resistance to blood flow. And as we learned earlier, the highest and lowest pressures in the cardiovascular circuit are in the ventricles during pumping. And so it's ideal that this circuit then can move blood throughout the entire system by generating this very high pressure gradient. What we also know from our Poiseuille's law and this resistance factor is that mean blood pressure falls as it moves through the peripheral and pulmonary circuits, and that these one-way valves are quite critical to this driving the blood in a unidirectional manner. If those valves weren't there, you'd have significant backflow during diastole into the heart. Now, Ohm's law is important generally in the cardiovascular system, so you've got this need for a gradient across the entire system, but it's also important at the tissue level as well. So what I mean by that is the different tissues need to be able to respond accordingly depending on their metabolic needs. So if we look at this diagram on the right-hand side, we see an overview, and let's say this is the basal state of how blood is distributed. And this is interesting in its own sake, just for you to go through and see how much blood or what proportion of the total blood flow is directed to the different tissues. But on top of that, for those of us who have had a meal or who are on the treadmill and we're exercising, we need to have sort of dynamic changes in blood flow between our gut tissue, between our skeletal muscles, in order to coordinate optimized blood flow to the tissues that need it the most. And these localized changes in blood flow are also mediated by the Ohm's law relationship. Okay, and so we've restated it on this slide that the pressure in the artery minus the pressure in the veins is obviously the delta pressure across the tissue, and that's going to determine the blood flow into the tissue and the resistance within that tissue, or at least they're all intimately related. Now it's worth going over an example of how these things may change at the level of the arterioles. So what role may an arteriolar bed have in determining the blood flow into a tissue? So if we look at the top example here, this will be our basal state example. We'll come from the aorta, which is pumping around 4 liters per minute, into 4 tissues. And you can see the blood flow is equally divided into tissues A, B, C, and D in this diagram, so that the 4 liters per minute is then distributed 1 liter per minute to each of the 4 tissues. Let's say, for example, there was a need to shut off blood flow to tissue B. How might this impact the other tissues? Well, we know from Poiseuille's law that if we increase the resistance in that tissue B, that we're going to have decreased blood flow, and that's exactly what we see in this lower example of the diagram. We squeeze down on the arteriole for tissue B, and we see that its flow drops to 1/4 of a liter per minute. But what's interesting in this example is that the entire system is still being fed at 4 liters a minute. And so what happens is tissues A, C, and D pick up the extra flow. And so now you can see, whereas previously they were at 1 liter per minute, now they're at 1 1/4 liters per minute to accommodate that extra 3/4 liter per minute. And this is exactly what happens in the physiologic regulation of different tissues. You squeeze down the blood vessels ahead of one tissue, and support increased perfusion across a number of other tissues. Another relationship that we need to think about is the number of pathways for blood flow within a tissue. So the number of pathways for blood flow actually also determine the resistance of that tissue at the capillary level. So you can see a very highly perfused tissue. So this is a metabolically active tissue in our example on the top of this slide. You can see blood flowing through all of the different capillary beds. And so this is a low resistance circuit. The blood has a number of different choices in where it can flow. And so the overall resistance of that tissue to blood flow is low. And obviously then you get increased blood flow as a result of that. If we look at our example on the bottom, we see that there are a number of capillary beds that are not filled with blood. This is because precapillary sphincters, or little muscle groups, can squeeze down in metabolically inactive tissue and prevent flow going into the capillary beds. The blood is then shunted through this central arteriole called a metarteriole, and under this circumstance, this metabolically inactive tissue is thought to have a high resistance to blood flow because there aren't a number of choices for the blood to take as it flows through the tissue. So metabolically active tissues are low resistance, metabolically inactive, high resistance tissues. And if you get a number of high resistance tissues across your entire body, then your total peripheral resistance, or your overall resistance, goes up. And that's an important concept when we start to think about blood pressure regulation globally. Before we get there, it's important to spend a bit of time in these capillary beds understanding a final sort of regulatory step in molecular distribution and blood flow, and that is the velocity of blood flow. So what we see on the top part of this diagram is the cross-sectional area of the different vascular beds. And so what I mean by that is that as the blood comes out of the aorta is only 2.5 centimeters squared. Okay? So that's all of the cardiovascular flow going through a fairly small diameter piping. As we start to branch out into all the different tissues, what you start to see is that the cross-sectional area, when you add all of those branches together, starts to go up dramatically. And by the time you reach the capillary beds, your cross-sectional area is actually about 2,000 times what it was when you started at the aorta. So you've got all of the blood going through a 2.5 centimeter squared tube or going through a 5,000 centimeter squared tube, and that makes a big difference to how fast that blood is moving at the different stages. So as blood moves through the vascular system, it slows down. And it's ideal that blood is at its slowest point in the capillaries because that's where the nutrient exchange has to happen. So you want optimized diffusion and very slow moving blood through the capillary beds at that point. And in fact, that's exactly what you see when you look at the velocity and cross-sectional area relationships. So while we're in the capillary bed, it's interesting to look at how materials are transported into and out of the capillary. And before we do this, we have to understand two concepts: filtration and absorption. Filtration is the net flow out of the capillary of solution and fluids. And absorption is the net flow into the capillary of fluids from the extracellular matrix. Across the entire capillary bed, it's interesting to note that the arterial side contains much more filtration and the venous side contains much more absorption. And this relationship is mediated by two very important pressures within the capillary: the hydrostatic pressure, that's trying to drive fluids and reagents out of the capillary, and the colloid osmotic pressure, which is the attraction of water to the proteins within the lumen of the capillary that's trying to bring fluids back in. And so we can look at those two relationships by comparing the red and the blue lines that are above the capillary image. The hydrostatic pressure, as you can see, decreases across the length of the capillary. This is because as the blood moves through the capillary, it's exposed to more and more resistance, And so the pressures drop accordingly. We've seen this across the entire cardiovascular system. This should make sense. So we move from 32 millimeters of mercury at the top to 15 millimeters of mercury at the bottom. Interestingly, the colloid osmotic pressure, that is that attractive force of the proteins inside the lumen, doesn't change across the length of the capillary. And this is because the blood proteins levels don't change from one end to the other. So the net result of this is that you have higher outward pressure from the capillary pressure at the arterial end, and that's driving your filtration, compared to higher colloid osmotic pressure at the venous end, and that's driving materials into the capillary. And this, from a physiologic perspective, works out very nicely because you want to deliver materials to the extracellular fluid at the arterial end, and you want to collect all the wastes and deliver them back to your sort of waste management systems at the venous end of the system. This is going to take us now to an understanding of blood pressure. And it's important to understand what blood pressure is, where it's measured, and why. So when we're talking about blood pressure, we're talking about the pressure inside the larger arteries. And that's actually where we measure it on patients. And that's actually where your body measures it. When your body's trying to sense, using these pressure sensors within the cardiovascular system, it's taking a measure of the pressure within those large arteries. Why is that? Well, this is the main source for tissue distribution for all your vital organs. Okay, so the pressure that's kept inside those arteries then determines how well all of your different tissues can be fed. So when we look at the circulatory system as a whole, it's very helpful to consider it as a system of fluid-filled bags. So what do I mean by this? We have an arterial artery system that is a low compliance, low capacitance system, which means it's a very stiff bag, and so it doesn't tend to hold a lot of blood. On the other side, we have the large veins, the vena cava, and that system is a high compliance, high capacitance system that actually has the capacity to hold large amounts of blood. And you can see this in the way that the total blood volume is distributed. 70 to 80% of all the blood is found on the venous side of the system, and only 15% is found in the arterial side. And that makes complete sense when you think of it as a series of fluid-filled bags. It's harder to fill a stiffer balloon for the arteries than it is for the veins. And so this is how blood normally distributes. Another key component of this is that as you increase the total blood volume, and this is the role of the kidneys that we'll get to later in this lecture, then you change the amount of blood in those bags, and you change the blood pressure accordingly. So by increasing blood volume, you can have an impact on blood pressure in the larger arteries and on the filling of the venous system. The last thing I want to point to is that these fluid-filled bags aren't in isolation, they're in the entire cardiovascular circuit. And so we have the heart in between the veins and the arteries, and we have the resistance arterioles between the arteries and the veins. Imagine what would happen if you increase the function of the heart and draw blood out of the veins into the arteries. You're actually going to increase the pressure in the arteries. Because they're so stiff, it takes only a small amount of blood to move from one bag to the next to get a large effect on arterial blood pressure. Likewise, the resistance arterioles are the conduit between the arteries and the veins. And so if you see a resistance vessel squeezing, that's going to prevent blood flow from the arteries. That's going to increase the pressure as the volume increases within that arterial bag. And so these are key steps for the regulation of blood pressure and cardiovascular function, and we're going to go into detail in some of the mechanisms of them over the next few slides. The first mechanism we're going to address is this idea of volume changes. And volume changes are linked to the slow response in the kidneys. there are two types of blood pressure responses: the slow response that we see on the right-hand side of this diagram, and the fast response, which is thought of as the autonomic response or the compensatory response by the rest of the cardiovascular system. So the kidney takes care of the slow end of things, and it determines what our total blood volume is over a period of minutes to hours. The fast response happens in seconds or sub-seconds to help us regulate very rapidly the blood pressure within the arteries. So the long-term regulation that we referred to are these volume control mechanisms, and the kidneys are key to this blood volume control. So changes in blood volume affect a number of different aspects of the cardiovascular system. So for example, changes in blood volume in the veins affect venous return to the heart that can ultimately affect how well the heart pumps or the cardiac output. Changes in blood pressure globally drive changes in pressure natriuresis. So this is the ability of the kidney to excrete more water when you've got higher blood volumes and higher pressures in the system. And so that's one of the important roles of the kidney, is when you've got high pressure, then it squeezes out more water so that that can be excreted. Kidneys also contain the machinery to produce a number of important hormones that are key to blood pressure regulation. And in a separate video, I discussed the renin, angiotensin, and aldosterone system and its role in blood pressure control. And so I'll save that for that video. There are a number of conditions associated with long-term blood pressure regulation. So if your blood pressure is dysregulated, you can have hypertension, which is high blood pressure. You can have hypotension, which is low blood pressure. And there's various forms of shock, distributive shock, that ultimately lead to life-threatening events. Volume control is one aspect of global blood pressure control. So it's the long-term aspect. I mentioned that there are also a number of rapid regulatory mechanisms that are controlled by these reflex responses of the cardiovascular system. And these reflex responses feed back to how well the heart works as a pump. Okay, and so this is determined by heart rate and stroke volume. It also feeds back to how the resistance vessels squeeze down on the various tissues. So the diameter of the arterioles is affected by these rapid compensatory changes. And then in addition to all of this, that floppy venous bag is also innervated by the sympathetic nervous system, and so the compliance of that bag can change as the smooth muscle cells start to squeeze on that vascular bag. And so under certain circumstances, changing the compliance of that bag can deliver more blood back to the heart and help the heart pump more effectively. So all three of these are integrated fast blood pressure regulatory systems. This fast or short-term BP regulation is basically controlling pumps and resistors. If we look at the artery as a sleeve of vascular tissue, if you increase the pumping factor but don't change the resistance factor, you essentially blow up the balloon, the pressure increases. That's exactly what happens at the level of cardiac output regulation. If we contrast that, if we keep cardiac output the same, but we squeeze down on those variable resistors, then we can also see a buildup of mean arterial pressure, a stretching of that arterial bag, increases in pressure. And so these are the two mechanisms that the body uses to control blood pressure in the arteries. And so we look at this equation, then mean arterial pressure is proportional to cardiac output and to the resistance of the cardiovascular system. The regulation of cardiac output, very simply, can happen at the regulation of heart rate and stroke volume. So we've learned in other lectures that heart rate can be controlled by the sympathetic nervous system at the level of the sinoatrial node. So we increase the number of contractions per minute. Stroke volume, because the ventricles are innervated by the sympathetic nervous system, they can be encouraged to pump more forcefully when you have high sympathetic activity. And this can increase the volume that is pumped per contraction. And so by activation of various sympathetic pathways, we can increase the cardiac output because it's proportional to stroke volume and heart rate. And as we mentioned previously, if we squeeze on that venous bag as a final way of increasing venous return, there's a law called the Frank-Starling law that tells us that the heart can essentially pump all of the blood that's returned to it. Increased stretching leads to increased contraction and increased cardiac output as a result. And so all of these factors together then build together to increase cardiac output in response to sympathetic activation. When we look at the vascular contribution to blood pressure regulation, we come back to our friend Pussoi. And Pussoi's law basically tells us that the resistance is related to the radius of the blood vessel. This looks like a fairly complicated equation, but if you look at it, most of these other factors come out as constants. The blood viscosity, the length of the tube, they're all constant in a blood vessel, and really the only thing that can change is the radius of the blood vessel. And it's quite remarkable, actually, that the resistance changes to the inverse of the fourth power of the radius of the vessel. So it follows that only very small changes in blood vessel radius can have a profound effect on blood flow. And so here you see reduction of the lumen radius by 1/2 can result in a 16-fold increase in vessel resistance. The sympathetic nervous system controls vascular resistance at the level of the arterioles. So the sympathetic nervous system, if we remember from our anatomy portion, interacts directly with the adventitia, and there are receptors called alpha receptors that live in the smooth muscle cells, and so norepinephrine coming from the sympathetic neurons rains down on these vascular smooth muscle cell alpha receptors. This leads to contraction, and as we see on the lower left, we get a smaller lumen radius as a result. So in this case, sympathetic activity squeezes the blood vessel, increases the resistance to blood flow. If we withdraw sympathetic effects, we get exactly the opposite, and that's what's shown on the right. You go from a blood vessel at sort of a steady state level to one that's completely vasodilated in the absence of the sympathetic activation. When we look at other feedback control mechanisms, there are these pressure sensors that live in the cardiovascular system, found mainly in the area of the aorta and the carotid arteries. And these pressure sensors feed back to the cardiovascular control center in the medulla oblongata. And this is where the sympathetic and parasympathetic activity is modulated. So if these baroreceptors sense an increase in pressure, for example, they fire a number of signals back to the medulla oblongata. At the cardiovascular control center, the increased pressure then is interpreted that you need to reduce sympathetic activity and increase parasympathetic activity. And so then those signals are then fed back to the key effector tissues of the sympathetic nervous system that relate to cardiovascular regulation. And those are the sinoatrial node. Okay, so in this example, higher pressures would lead to higher parasympathetic activity or a slowing of heart rate at the sinoatrial node. Likewise, you feed back sympathetic information to the sinoatrial node, you feed it back to the myocardium, you feed it back to the veins, and you feed it back to the arterioles. And in the case of higher pressures, you would have less sympathetic activity going to the sinoatrial node. That would slow heart rate. Less sympathetic activity going to the myocardium. That would give you decreased stroke volume. Less sympathetic activity to the veins, and so that would make them more stretchy, less and more compliant. And then you'll have less sympathetic activity to the arterioles, and those are that are regulating the flow out of that arterial bag. And so as a consequence of all of these events, you're going to have a feedback control system that ultimately lowers blood pressure in that arteriolar bag as a result of sensed increased pressure. One of the last things we want to look at is something that can happen in individuals when blood pressure goes wrong. An example of this is orthostatic postural hypotension. I actually have personal experience with this because my younger boy, if he gets up out of bed too fast, will often faint or fall down. And so what it really means is that if you stand up too quickly, you start to feel lightheaded, you get blurred vision, or you have fainting spells as a result of this. And the reason for this is because as we stand up, gravity pulls blood away from the heart down towards the extremities, and you have less blood in the veins that's capable of returning back to the heart. And so individuals that are already hypotensive, and I think this is the case for my son, his blood pressure is very low, or don't have strong baroreflex mechanisms that can restore the pressures or the arterial pressures back to normal levels, then get reduced blood flow to the brain and as a result have a fainting spell. And so this is a sort of clinical example of how baroreceptor function and integration of blood pressure with the baroreceptors and the rest of the cardiovascular system is very important. So I want to thank you for your attention and just sort of reiterate that by now you should be able to describe the determinants of blood pressure and its relation to vascular resistance, and in addition understand the autonomic control of blood pressure regulation in the cardiovascular system. Thank you very much.